All right, welcome to segment 2. We're going to talk about the details of the overview of basically cell division that we discussed in the first segment. So if we look at this, remember we start out with a parental cell or a parent cell, and this one is very simplistic. It only has one chromosome. You can see the chromosome does have a long arm and a short arm. Now all of them have that. And there's a centromere. Right? So remember, the centromere is where it's kind of constricted. Now, what happens before the actual division of the cell, so this is in preparation, the chromosome is duplicated. So the chromosome is duplicated, and once the chromosome is duplicated, now you have two identical copies of this chromosome. We could call it chromosome 1. They are connected together by a bunch of proteins, okay, in the same manner as each other, right? So just basically adjacent to each other. They are now called sister chromatids. Okay, so this whole thing is a mitotic chromosome. However, the mitotic chromosome is made up of sister chromatids, and these are identical copies of each other, and we're going to just call this chromosome 1. Well, after nuclear division and cytokinesis, we end up with two daughter cells, which are identical to the parent cell, or essentially identical to the parent cell, each with one chromosome. So here they're called sister chromatids, but after cell division, they are now individual chromosomes. Now let's go through the details of the cell cycle. Eukaryotic cells pretty much alternate between two major phases. That's interphase and M phase. Now, cytokinesis is completely separate from mitosis or mitotic phase, but it does overlap with it, so it's kind of part of that phase. Now, the cell is going to be in interphase the majority of the time. It's probably about 90% of the time the cell is going to be in interphase, and a lot happens during interphase. Right, so the interphase is divided up into 3 subphases. That's G1, S, and G2. So G1 is the gap 1, and G2 is gap 2, and then S phase, the S stands for synthesis. So during gap 1 phase, the cell has just undergone cell division, and it's in the process of growing. Now, if it passes a checkpoint, which we'll talk about later, then it will enter S phase. Okay, S phase is when the chromosome is duplicated, like we just discussed in the overview. And then after the chromosomes are all duplicated, the cell will enter G2 phase, which is really the preparation for M phase. So it's during G2 that all of the centrosomes are duplicated and a lot of the proteins are duplicated. So it's in like maximum growth stage there. Once it passes G2 phase, it enters M phase. And M phase and cytokinesis kind of overlap with one another. Now, if we put this on like a clock, okay, so we can imagine that we have a cell, and this pie chart represents a clock, or the time it is in each of the phases. So we could just start here where, let's say, this cell, A cell, has just undergone cell division. Okay, so now we have 2 copies, right? So we have 2 daughter cells, which are identical to one another. This cell may undergo another division, but we're going to follow this cell. Okay, so this cell is going to immediately enter G1. It's going to pass G1 if it's meant to, right? It's going to pass G1, and there's a checkpoint here, and it's going to enter S phase. And then S phase is when the DNA is duplicated, and then G2, like I said, this is where most of the growth and preparation for mitotic phase occurs. Now you can see the majority of the time the cell exists is going to be in interphase, and then a very small amount of that life cycle of the cell is in M phase, which includes mitosis and cytokinesis. Now, M phase is further divided up into sections. Right? So it's broken down typically into about 5 stages. This includes prophase, prometaphase, metaphase, anaphase, and telophase. What follows this is cytokinesis, and cytokinesis really kind of... Okay, so let's start at G2. Remember, G2 is the last subphase of interphase. So looking at the pie chart, we're right here, and it's just before the start of M phase. So during G2, the centrosomes are duplicated. You can see them kind of highlighted a lot darker in this particular image because there's two of them now, right, during this stage. And you can see both of them here. Okay, so the centrosomes are duplicated. And at this point, all of the DNA has already been duplicated. So there's 2 copies of each chromosome present here. The next phase, which is going to be part of M phase, which is called prophase. So here we have G2, which is what we just talked about. The 2 centrosomes have been duplicated. Now the centrosomes are going to start to move to opposite ends of the cell, and the mitotic spindle is starting to form. The mitotic spindle includes several components, which we're going to talk about the details later. However, just briefly, we have 2 asters which are going to form and some microtubules which are going to extend from each of the centrosomes where the asters are. So remember, the centrosomes are each made up of 2 centrioles. Right? And this is the microtubule organizing center. So the microtubules, remember, are made up of the protein tubulin. And tubulin starts to polymerize and it creates these long structures, the long microtubules. So again, they're going to extend all the way from each of the ends once the asters and the centrosomes, right, with their centrioles are on complete opposite ends of the cell. Also during prophase, the chromosomes are starting to condense. So you can see them much more condensed in this image here, and you can see the centrosomes starting to separate. Now we're going to compare, we're going to look at what prophase looks like. So remember, plant cells are more rectangular or square in shape, and you can really see a defined cell wall around here. These are also stained, so you can see the chromosomes a little bit better. Here we've got the nucleolus. Remember, the cell is undergoing cell division, and it's going to need a lot of ribosomes. Remember, that's where the ribosomes are synthesized. So it's going to need a lot of ribosomes, so the nucleolus is going to be pretty evident. We have chromosomes which are condensed, and of course this makes up the nucleus. Now, in plant cells, there's no spindle that's formed. It's called a phragmoplast. Now, there are microtubules in which there's vesicles that are going to travel along the microtubules to eventually divide the cell. And we'll talk about cell division in plants and animals and the differences between those in just a little bit. All right, so that's prophase in plant cells. Now let's compare prophase with prometaphase. All right, so in the animal cell, we can see here the fluorescence micrograph, and then we have the prophase, which we just talked about, and now prometaphase. At prometaphase, the centrosomes should be on opposite ends of the poles, and the microtubules have grown, so basically the mitotic spindle is just about done forming. What happens during prometaphase is the microtubules are either going to attach to the chromosomes or they're just going to extend and kind of associate with ones on the opposite end. The ones that attach to the microtubule or to the chromosomes are called the kinetochore microtubules because they bind to a kinetochore, which is basically a complex of proteins at the centromere of each of the chromosomes. Okay, so the kinetochore microtubules are going to bind to the kinetochore, which is at the centromere of each of the chromosomes. And then the non-kinetochore microtubules are going to extend as far as they can and associate with other non-kinetochore microtubules from the opposite end of the cell. And you can see that here. Again, the chromosomes are further condensing. They're almost completely condensed at this point. So they're further condensing. You could see the centrosomes on opposite ends, and you could see the kinetochore microtubules if you look close enough that they are connecting with some of the chromosomes. Okay, so in the plant cell, it's going to be pretty evident where the chromosomes are going to be quite a bit darker, right? And again, you're really not going to see the centrosomes because there isn't any centrosomes. Okay, so cell division occurs a little bit different in plants. However, you can see that this is the prometaphase because the nuclear envelope isn't as defined and the chromosomes are a lot more condensed. Now, the next stage is pretty obvious when it occurs. It's called metaphase. So here's prometaphase. Here we can see the animal cell, the nuclear envelope is degraded. It's not present anymore at prometaphase. By metaphase, it's completely gone. Now, at metaphase, all the chromosomes line up at the equator of the cell. And this creates an imaginary line called the metaphase plate. Okay, here the spindle has completely formed. Okay, so the mitotic spindle is already set up to go. Okay, so again, the centrosomes are going to be on opposite ends, and the microtubules are going to be extending as far as they can. We have those that are connecting to the kinetochores of the centromeres in the chromosomes, and some that do not connect. So that is the difference between the kinetochore microtubules and the non-kinetochore microtubules. And you can kind of see that up here. You can see all the chromosomes, even they're not going to line up. This is just a cartoon picture right here, but this is what you would see. You can kind of see that imaginary thick line of chromosomes lining up at the center of the cell, right? The equator of the cell. And then you could see the microtubules extended with the centrosomes on either end of the cell. What it looks like in plants. So we can see there's no nuclear envelope. It's completely gone. And you can kind of see that very dark imaginary line called the metaphase plate when the chromosomes line up at the equator of the cell. Now, it's really not the equator. It's really the center of this rectangular cell. An equator is mainly for a spherical type cell. Now we're going to compare next phase, which is anaphase. So remember, in metaphase, all the chromosomes are lined up at the equator. Well, then what happens is all of the chromosomes, basically the sister chromatids, are separated from one another. And how that occurs, and we're going to talk about the details later, is that the microtubules start to depolymerize. And at the kinetochore, there are motor proteins that walk along the microtubule. So basically, each of these sister chromatids are going to be pulled apart from one another during this particular process. So you should see a space in between these, here it's calling them daughter chromosomes. They're separated now. They're not really sister chromatids anymore, right? So they're pretty much individual chromosomes. And you could see at this fluorescence micrograph, right? So you could see that the centrosomes are on opposite ends. You can see the microtubules are still attached to the sister chromatids, which are being separated because you can see this line of clearance in between the chromosomes. So they're being pulled apart, like in a plant cell. Right, so they're still very condensed, right? There's no nuclear envelope present, and there's some space that's kind of developing in between about equal halves of the chromosomes. And then the is telophase. However, cytokinesis kind of overlaps with it. So remember, anaphase is separating the sister chromatids. Well, by telophase, now this is advanced telophase because you can see cytokinesis is also occurring, but basically the chromosomes are going to start to decondense. The nuclear envelope is going to start to reform, and there's, in animal cells, a cleavage furrow is going to form. A cleavage furrow forms by the constriction of part of the cytoskeleton, which we'll talk about in just a moment, and it pinches off the cells so that it'll end up two separate cells, each of them having only one centrosome and one nucleus and an equal amount of chromosomes. And this you can kind of see in the fluorescence micrograph. The cells kind of separated. You can kind of see the cleavage furrow in these types of animal cells. And then they're going to end up with equal amounts of DNA as long as each of the sister chromatids were separated to each of their individual cells. Looks like in a plant cell. Now in a plant cell, a cleavage furrow doesn't form. A cell plate forms. Remember, I was telling you that the plant cell, remember from chapter 6, does not have the centrosomes. They have a different type of system, and what develops is called a phragmoplast. A phragmoplast is basically a network of microtubules which allow vesicles. Remember what vesicles are? They're like membrane bound spheres which move around inside the cell, where they're going to travel along these microtubules to the center of the cell, and they're going to start to line up there. And this is what the cell plate is starting to form. Eventually, those vesicles are going to fuse with one another. Once they fuse, they form cell membrane, and then the cell wall can develop around the cell membrane after that. Right, so let's kind of watch a little bit. Let's watch a little bit of this happening. Okay, so you can see this is movement, right? So we've got the chromosomes, they're condensing and they're moving around. Oop, they're starting to line up. All right, look at that. Okay, look at, so they're lined up. Whoa, and they're separating, look at that. And now the cleavage furrow. Now, let's look at the mitotic spindle a little bit closer. We're going to talk about the Pac-Man mechanism as well. So remember, by metaphase, the mitotic spindle is fully formed. So if we look at this cartoon picture, we can see the metaphase plate. All the chromosomes have lined up at the equator, and the mitotic spindle is fully formed. What the mitotic spindle includes is the two asters, one on either end. The aster includes the centrosomes and some shortened microtubules which help maintain the shape of the cell. And then the centrosomes, of course, what's extending from each of them are going to be the microtubules. Now remember, there are kinetochore microtubules and non-kinetochore microtubules. The kinetochore microtubules are going to be the ones that bind to the kinetochore, which is a set of proteins at the centromere of each of the chromosomes. You can kind of see that here, this transmission electron micrograph, right? So we can see the kinetochore and the microtubules attached to it. And this shows another micrograph here. We can see metaphase and the microtubules with the centrosomes and the asters on either end. And then, of course, the kinetochore microtubules and the non-kinetochore microtubules. Now let's talk about how the if we look at this right here, we have the spindle, which is fully formed, and we can see that the cells are starting to undergo anaphase. Now, what happens at anaphase is there's a set of proteins called cohesins, which keep the sister chromatids together. Well, there's an enzyme called separase. It's named according to its function. So basically, it cleaves the cohesins so that the sister chromatids can separate. So the sister chromatids will easily be able to separate once the microtubules start to depolymerize. So remember, I told you tubulin starts to polymerize when they're forming. However, it's going to depolymerize when they're starting to get short. Okay, so that is that process there. So let's look at it closer. So looking at this, we can see this is the process of anaphase. What's actually happening at the kinetochore is the following. So here we have the sister chromatid. And then we have the kinetochore, we have proteins here. Part of those proteins involve a motor protein. Remember, motor proteins kind of walk along the microtubules. Well, this motor protein is walking this way. You can see where it says chromosome movement. So the chromosome is moving this way along the polymerized part of the microtubule. While on the other side of the chromosome, the microtubule is depolymerizing. So it's breaking down into its individual tubulin subunits. Okay, so that's how that's happened. So basically, as these chromosomes are moving along the microtubule toward the opposite ends, on the other side of the chromosome, the microtubule is starting to depolymerize or break down. Right, so let's talk about cytokinesis a little bit closer. Okay, so let's compare cytokinesis in animal cells with plant cells. So this shows cleavage of an animal cell. This is a scanning electron micrograph of an animal cell forming a cleavage furrow. What actually happens is the microfilaments, not the microtubules, these are microfilaments, they form a contractile ring, and then it contracts, and it basically pinches off the cells. Okay, so this happens in animal cells. And in plant cells, I kind of touched on this, right? So we have a transmission electron micrograph showing the inside of the cell and the vesicles forming this line along the center in between the two nuclei, right? So the nuclei are divided here. Now the cell plate forms. And what this is vesicles. They start to build up at the center of the cell, and then they fuse together, creating this cell plate. This is made out of membrane, right? So the membrane eventually fuses with the outer part of the membrane of the entire cell and then divides it up into two cells, then the cell wall can form. So again, there's a difference between animal cells and plant cells. In animal cells, a cleavage furrow forms and they are pinched off. In plant cells, a cell plate forms due to building up of plasma membrane in between the two nuclei. All right, let's talk about bacteria. So we're going to talk about eukaryotic cells, which are quite complex. Bacteria do undergo division, right? Except it's called fission, and it's a lot simpler than what happens in a eukaryotic cell. Bacteria, basically, remember the bacteria have a cell wall. Of course, they have a plasma membrane that surrounds the rest of it, but they don't have a nucleus. They just have a chromosome in there. Well, the chromosome is replicated just like what happens in the eukaryotic cell, except replication happens a little bit differently, which we'll discuss when we get to chapter 16. But ultimately, there's what's called an origin of replication that kind of begins right there, and you can see this like bubble forming. Well, that's the bubble which is formed at the origin of replication once replication begins. Now, the chromosome is basically duplicating, right? So basically they're going to kind of separate. Not a whole lot of detail is talked about in your textbook. So basically you're going to have two chromosomes, one on either end of the cell is going to start to separate. Okay, so it's going to start pinching off. However, it's not like what happens with the eukaryotic cells because there are no contractile rings that form because there's no cytoskeleton in these types of cells. However, it does divide, and again, it's similar to mitosis in that these are identical daughter cells to the parent cell. However, it's not as complicated as what occurs in the eukaryotic cell. All right, so in terms of the evolution of mitosis, there are some thoughts about that, of course. Now, we know that prokaryotes evolved before eukaryotes. So mitosis probably evolved from binary fission. So remember, prokaryotes undergo binary fission. So basically, the process of binary fission was probably changed over time to accommodate eukaryotic cells and resulted in the more complex stages of mitosis. Some protists exhibit cell division that is kind of in between binary fission and mitosis, right? So it's kind of in between there. And protists are one of those groups of organisms, right, that evolved sometime after prokaryotes, but before, the more complex eukaryotes like us. This kind of compares some. So we have the bacteria, right? And then we have the eukaryotes, which is this. And then this shows like the ones that are kind of in between, right? So we can see the dinoflagellates have microtubules, right? You can see the chromosomes, they're separating in some way, and they have a nuclear envelope. Now, the diatoms in yeast show similar things, right? So we have kinetochores with microtubules as well with a nuclear envelope. So it occurs somewhat differently, and it looks like it's somewhere in between the bacteria fission and the eukaryotic mitosis. So that kind of, explains a little bit about the evolution of mitosis in general. So this is basically a summary of this particular segment. I went through the detailed stages of the cell cycle and the stages of M phase, and then we talked a little bit about how bacteria replicate or reproduce, and a little bit about the evolution of mitosis, at least the, the theories behind it. Okay, well, that's it for this segment. Thank you.